Hello, good afternoon. Welcome to the webinar, Advanced in Gene Editing and mRNA Vaccine Technology in Brazil. This symposium is promoted by the American Society of Gene and Cell Therapy with the Brazilian Association of Cell and Gene Therapy. Firstly, I'd like to thank all the invited speakers for accepting to participate in this symposium. Thank you very much. The main goal of today's program is for the speakers from the US and Brazil to come together and share their expertise on vaccine technology and gene editing. Speakers will present and discuss the impact of mRNA technology on the vaccine field and the challenges and successes in manufacturing and viral vector design. The latter half of the event will focus on in vivo and ex vivo gene editing from basic science through clinical development. If you have any question, please type questions into the Q&A throughout the presentation, and you may continue to submit questions throughout the panel discussion. Questions in each section will be saved until the panel discussions. This is the second webinar between the American Society and the Brazilian Association of Cell and Gene Therapy. The first webinar, which was held about 2 years ago, and one of the topics of that webinar was CAR-T-based gene therapy. Today, I'm very happy to announce that the Brazilian FDA called Anvisa has just approved the first CAR-T-based drug for leukemia. And I'm very proud to say that the associated members the Brazilian Association have participated actively in the evaluation of this and other two gene therapy products, Zolgensma and Lusterna. This achievement demonstrates how important is such a symposium, speaking the issues of public and scientific interest. So thank you for your attention. Now I invite Dr. Martin Bonamino to chair the first session.
Thank you very much, Dr. Sang, and thank you to the ABTCel-Gen and ASGCT for organizing this very nice event so we can discuss about these topics that are wide interest. And we will start with the mRNA vaccines. It has been a big novelty. It's a large-scale implementation the last couple of years, and we will have two speakers today. So the first one will be Patricia Neves, who is the project manager and scientific leader of the mRNA vaccine hub implementation for WHO at Biomanginius Fiocruz here in Rio de Janeiro. And Dr. Neves will be speaking on mRNA technology development for the World Health Organization. The World Health Organization set in some hubs for mRNA vaccine, and one of them will be at Fiocruz, and Patricia will be the person in charge. So she will speak about the clinical experience and production for these vaccines. And following Dr. Neves, we will have the talk from Dr. Margaret Liu, who is a physician and researcher studying gene expression, immune response, and vaccines. She was previously the president of the International Society of Vaccines and is currently the chairman of the board of the ISV. Dr. Liu will discuss nucleic acid vaccine technology for both mRNA and DNA. So mRNA and DNA-based vaccines. We will broadcast both of the presentations, and then we will, soon after the presentation, we'll have the Q&A session. So please, if you have any question, just put it on the chat. Thank you very much, and I'm looking forward to our discussion session after the presentations.
Hello all, my name is Patricia Neves. I'm the RNA Vaccine Hub Project Manager here at Biomanginius and also the scientific leader of Hub implementation. I will talk a little bit with you about Biomanginius as a hub for mRNA technology development, for who our clinical experience and production. So last year, WHO, together with PAHO, asked for expression of interest, aiming to establish hubs for mRNA vaccine development and production in Americas. The key points from HUD for these hubs development and implementation were asking for small and middle-sized manufacturers which could host a COVID-19 mRNA hub, and owners of technology interested in transferring to enable production of mRNA-based COVID-19 vaccines in low and middle income countries. So, as Biomanguinhos was developing our own technology at that time, we submitted to this call and we were chosen by WHO and PAHO for composing a hub with Argentina here in Americas and also with Afrigen in Africa. Talking a little bit about Biomanginius Technology, as you may know, there are two different types of RNA vaccines. The conventional RNA, where the RNA is directly directed to the cytoplasm of the cell, and then it's directly translated, expressed protein that we are interested in. So there is no amplification of the conventional RNA. This technology is like the Pfizer and Moderna's vaccines. On the other hand, we have the self-amplifying RNA technology, which is our technology. RNA construction has a viral replicase machinery, and then when the RNA is delivered to the cell, we have cycles of replication of the RNA, amplifying the antigenic mass after application. This technology has enhanced and prolonged antigen expression. We need lower effective RNA doses, potentially resulting in better safety. We have an intrinsic adjuvant effect and potential apoptosis of vaccine-carrying cells due to the vaccine self-amplification leads to enhanced cross-presentation, which is very good for T-cell immune responses. And for us, the most important characteristic of this technology is the option for single vector delivery of multiple or complex antigens. Like is explained here, the self-amplifying RNA allow us to include a lot of antigens code in line under the control of subgenomic promoter. So it's much better to deliver the multiple and complex antigens. So we developed here at Biomanguinhos a proprietary plasmid and formulations totally designed by us. It's a plug-and-play technology. We could put in and take off wherever we want, the gene, the antigen we need. It's a self-amplifying technology. And for COVID-19, that we'll talk a little bit with you in the following slides, we have three different constructions engineering SARS-CoV-2 antigens to the desired cellular compartment. Our first target is spike protein, which is being used broadly in vaccines that are currently in market. Using spike protein, we aim to elicit the neutralizing antibodies, which have been implicated in protective responses, despite the fact that until now we could not achieve sterilizing protection. But spike is very broadly used in all vaccines we are using now. The second antigen we are working on is nucleocapsid protein. This is a very highly conserved protein between SARS-CoV-2 variants, and protein has been implicated in eliciting broadly and strong cellular T-cell responses. So by using two different antigens in the same construction, our vaccine for COVID-19 is considered a second generation vaccine. Self-amplifying RNA technology has also manufacturing advantages. This is a modeling study conducted by Imperial College of London, which points to lower costs for expenses and investments. As you can see here, compared with conventional mRNA vaccines. This model is based in what is being used at the market now and what's being studied in clinical trials for this kind of vaccines. And also we could see that lower, the scale of production is much lower in self-amplifying RNA technology than in conventional mRNA technology. So in terms of manufacturing, obtaining RNA is much more simple than working with cells. We just need the GMP plasmid obtaining. This model plasmid DNA is linearized, and with a few steps of using just control of temperature and vortexing, and use enzymes free of cells, we perform the transcription and capping steps. So the RNA goes to the formulation and encapsulation in order to protect the RNA from degradation, and then the sterile fill and finish. The advantage of this kind of manufacturing is that the nature of RNA never changes. So this this platform is perfect to give us very quick responses for future emergencies. We are preparing Biomanginius also to manufacturing RNA. We are equipping our facility for manufacturing. We have a 500 square meters area with capacity of 100 liters of IVT reaction to obtain the RNA. We are also working on quality control and quality management from our data, our experimental data, and working on process for KBD modeling and production process in order to accelerate our regulatory approval. So we are getting more prepared to work with RNA molecule. During the pandemic, clinical studies were incredibly accelerated, and the time to market reduced dramatically. We are expecting that the lessons learned during this period will be maintained, at least in part. So we could comprise the initial phase and see and collect real world data and accelerate also the next necessary clinical trials for other future vaccines. For our COVID-19 vaccines, we are expecting that we could perform our gold standard clinical trial, which is large scale, randomized and controlled, and double blind trial, but in a scenario where almost all people here in Brazil are already vaccinated, we could establish a correlate of protection. Until now, we are working in terms of neutralizing antibodies. And if it's confirmed, and now we could perform studies on seroprotection or serological non-inferiority. For Biomanguinhos formulation, we are expecting to start phase 1 clinical trials until the second semesters of 202. In terms of technology, mRNA clinical trials are trending up. So we are expect very good growth of this technology use. And some analyses are pointing that mRNA vaccines and therapeutic market will increase incredibly in the next 5 years, raising from 9 to more than 15 billion up to 200 to 26. Biomanguinhos as a hub for mRNA vaccine development, production, and quality control are being prepared inside all these areas, R&D, QC, regulatory. We are already designing our clinical trials and also our manufacturing area in terms of preparedness to be to receive this and develop this technology and to meet the Brazilian needs inside RNA technology, not only for vaccines, but also for therapeutic molecules. And we expect also to attend other countries, other low and middle income countries in the next few years. So thanks a lot.
Thank you very much for the invitation to speak. My name is Margaret Liu. I was asked to speak about mRNA vaccines, but I decided to expand it to include both types of nucleic acid vaccine technologies, mRNA and DNA vaccines, and you'll see why during my talk. I have a couple of disclosures. I'm a longtime member of the Oxford Jenner Institute SAB, which developed the AstraZeneca COVID-19 vaccine, and also have been a member of the WHO drafting groups responsible for guidelines for both DNA and mRNA vaccines. Of course, as you know, nucleic acid vaccines differ from traditional vaccines that instead of using a virus or a viral protein, they use the DNA or mRNA that codes for the protein. So while the drug substance is what's administered, the antibodies and of course T-cell responses are still directed against the antigen that is the viral protein. Now, there are many applications for gene delivery via nucleic acid vaccines. And so I'll just, of course, be talking about the vaccines, but as you know, these can be used as well for immunotherapy and for other gene delivery technologies that you'll hear about later, such as the gene editing. Now, I want to also further expand my talk from mRNA and DNA vaccines and the future to include One Health rather than just focusing on COVID-19. One Health recognizes that the health of people, animals, and the environment are intertwined. And so a comprehensive approach to health is needed, particularly for vaccine development. And of course, this is because diseases spread in both directions, from animals to people and people to animals. Now, those of you in Brazil, of course, are very aware of all this because Zika, of course, first arose with this sylvatic cycle within non-human primates in Africa, but then spread all the way around the world and then became largely a problem of an urban cycle with people. The organization of my talk is that I will first talk about the mRNA and DNA vaccine technology, describing their immunological mechanisms and other characteristics. I'll also talk about some of their limitations and areas for improvement. And then I'll talk about what to predict from past successes for future applications. Here in schematic form, you can see the composition of the DNA and mRNA vaccines. So the plasmid DNA vaccines are simply that, a plasmid where the gene encoding the antigen has a promoter so that they can be expressed and turned into proteins in the mammalian cell after in vivo delivery. These are given simply in a saline solution. In contrast, mRNA needs to be formulated, and the types of formulations that are now used for the COVID vaccines are lipid nanoparticles. Well, why were people so interested in these nucleic acid vaccines? There are many reasons, but the first was that there can be made very rapidly into new vaccines. So instead of having to spend all the time developing a cell line or growing the virus, you can simply, once you know the antigen and you know the sequence of the gene for the antigen, you can then design the vaccine very quickly. In addition, you can reuse the technology either for boosters or for new vaccines. And this is in contrast to viral vectors, where there is concern that anti-vector immune responses may limit the reuse. You may be able to use it for a boost, such as with the AstraZeneca vaccine. However, if one thinks about trying to make multiple vaccines with the same vector, there would be concern about limitation of the ability to reuse that same vector. Another advantage is an immunological one, because the body is what makes the antigen in situ. That means that the gene, either the mRNA or the DNA, can code for any kind of protein, whether it's a transmembrane protein or a soluble protein. And of course, if you're making a recombinant protein, you have the issue that you generally need a soluble antigen. And plus you have to worry about the post-translational modifications, such as glycosylation being the same as what would happen in a mammalian cell versus in the yeast or in the insect cell that makes the recombinant protein. A really key issue is that gene-based vaccines in general can generate T cells, and not just CD4 helper cells, but the MHC class I restricted cytolytic T cells, which are the CD8 cells. And this is important for responses to some pathogens, such as malaria, and probably even for most viruses, but also for cancer. And it certainly is important because since T cells can respond to epitopes from conserved antigens and not simply the outside proteins of a virus, that these CTLs may be able to contribute to cross-strain or superstrain protection. So this would be important because of, for example, the variants for COVID or for influenza. Now, I will be able to make my slides available and you can look at this later on the YouTube channel. So I'm not going to go through the details on this slide, but I just wanted to point out this table that came from a paper that I recently wrote with a colleague, Jeff Ulmer, that describes the evolution of nucleic acid vaccines and describes how many of the studies done from an immunologic perspective with DNA vaccines help to speed along the development of mRNA vaccines. But I also wanted to highlight that, in fact, the preclinical immune responses with mRNA for cancer were already shown in about 30 years ago. And so this is to highlight then what I'm going to be talking about next, which are the challenges for how to have made these successful RNA and DNA vaccines. So as you know, there are mRNA vaccines authorized and licensed only for COVID-19, not for other diseases, but obviously this has had an outsized impact and benefit for protecting the world because of the pandemic. For DNA, there actually is an authorized COVID-19 vaccine that it was authorized in India, but there are also a number of licensed products for veterinary use, both for vaccines as well as for non-vaccine therapies. I wanted just to mention a little bit about the DNA COVID vaccine, because one of the key features of this is that it uses a needle-free device. So this injector is a simple spring-driven device that was pre-qualified by the WHO and also even has marketing clearance by the US FDA for a number of years. So it's been used in the US for influenza vaccines and has been used worldwide for various vaccines, including the inactivated polio vaccine. So why did it take 30 years, almost 30 years, between those initial publications and then making the COVID vaccines? Well, one of the first challenges was that with mRNA, you needed to increase antigen production. And the reasons for this were that the stability of mRNA is very poor. The mRNA degrades very rapidly because of ubiquitous RNases. So different companies have solved this in different ways. You can use modified nucleosides, or you can sequence optimize the mRNA itself. Now, it would also be nice to increase the amount of protein, in this case, the antigen that's translated from the mRNA. And again, you can do this by codon optimization, which has been done for DNA vaccines as well, because you try to match it with the tRNA frequency in mammalian cells. Another technology is using self-amplifying RNA, which also increases the duration of the production of antigen. And I'll explain how this works in just a bit. This schematic shows the modifications of the in vitro transcribed mRNA that you can see that occur in the different parts of the mRNA. And self-amplifying mRNA differs in that rather than having the mRNA simply encode your antigen, it also encodes a series of proteins that comprise what is called the replicon that is in other RNA viruses. And what this replicon does is it enables then the cell that has been transduced to make not just have the one copy of mRNA that's involved in your mRNA molecule, but it actually then self-amplifies this mRNA molecule so that you make multiple copies of the mRNA coding the gene of interest, GOI, or the antigen. And therefore you can make a lot more antigen, and therefore you can at least in preclinical models, you can get a lot more potent immune responses based on having just per molecule of this mRNA. Well, mRNA has other activities, and in particular, it is a very potent stimulator of the innate immune system. So the in vitro transcribed mRNA stimulates several toll-like receptors as well as binding to other molecules, so that it has a net effect of causing a lot of inflammation as well as having impact on RNA degradation and RNA replication, etc. So one of the real challenges was to figure out how to use this mRNA in a way that would optimize immune responses, because you wanted to decrease the inherent undesired types of immune stimulation. And you, at the same time, though, wanted still to have the desired immune stimulation. So I'm sure you're all aware of the use of modified nucleosides, which was done to decrease the amount of undesired stimulation. But you can do this as well by using sequence modifications of the mRNA. And then in order to maintain the desired immune stimulation, the reason for this is that if you had, for example, a recombinant protein, if you gave that as the vaccine, while you would get some immune response, you really need to add an adjuvant to get an optimal immune response. So finding the right balance between these two has been one of the key challenges for mRNA vaccines. But as you can see, it's not the only challenge. So although a lot of attention has been paid to using the modified nucleosides, really the issue of formulation was just as big of a problem, because even that modified nucleoside mRNA alone would not have been successful for a vaccine, because you needed to formulate it to protect the mRNA and yet have the mRNA still be able to be delivered to cells that can produce the protein. And then once it's inside the cell, it still has to get out of the endosome into the cytoplasm. So many formulations have been tried, but the only ones that are currently used in the two mRNA COVID vaccines are lipid nanoparticles. And it's important to note that the different groups use different lipid nanoparticles. Well, another problem and another challenge that had to be overcome was manufacturing the mRNA vaccines. So back 30 years ago, actually, this was a really expensive process, even more expensive than today, that involved using rabbit reticulocyte lysate. And so to develop the manufacturing process where this could be done by simple in vitro transcription required a lot of effort to develop it. And then in addition, once you have the in vitro transcribed mRNA, that of course has to be formulated. And so the different lipid nanoparticle specific chemical combinations and the way of making them in a manufacturing process had to be figured out. And then we come to the other challenge, which is the temperatures for shipping and storage of the licensed and authorized mRNA vaccines. And that is that the mRNA still needs to be kept ultra cold. And so it's shipped frozen and then is stored frozen, except for just the very last bit of time. And in contrast, DNA is actually much more stable. It's stable at 25 degrees Celsius for over 3 months. So these issues are listed all here that had to be solved for making the mRNA vaccines for COVID, but they're still relevant to the future use, and I'll talk about them individually. So the first actually is delivery. And of course, it's actually very exciting that the DNA vaccine can be delivered by a needleless injector because of the issue that many people are scared of needles, as well as the issue of medical waste. From a stability standpoint, the DNA is more stable. And as we just said, the mRNA requires expensive freezers. However, it's important to note that certain mRNA technologies may be more thermostable, such as the CureVac vaccine that unfortunately had less than 50% efficacy in the clinical trials. But nevertheless, I think that different technologies are promising for making new mRNA vaccines that may be more thermostable. Now, I'm not going to really discuss this last issue of the post-injection stability, but from an immunologic perspective, there is probably an advantage to having the antigen persist for some period of time, because for the B-cell antibody responses, you still need to have somatic hypermutation. And so you do want the antigen to persist for some time in order to develop the optimal high tight binding antibodies. So from a manufacturing perspective, one of the real advantages of mRNA is that it can be made without the use of biologicals. So it's essentially a chemical process. However, this comes at the cost of being expensive to manufacture versus the inexpensive DNA. From a regulatory perspective, DNA is considered a platform technology by WHO guidelines. What this means is that once you make your plasmid, that if you use a different insert for a different vaccine, because then the only thing you've changed is the insert, that is considered then that particular plasmid, and delivery in saline would just be a platform technology. mRNA is a little bit more complicated because depending on what the mRNA sequence is, and therefore the length and the interactions with the lipid nanoparticles, or if you use different formulations or different lipid nanoparticles, then those wouldn't necessarily be considered to be the same vaccine in the sense of being a platform technology. From the perspective of global access, we've talked about how the generic manufacture of DNA versus the proprietary nature of not just the mRNAs and lipid nanoparticles, but of the actual manufacturing process is a challenge for global access for poorer countries, as well as then just the simple cost of the manufacture and the infrastructure requirements. Now, one of the issues that I think we're starting to explore even more now is the immunization protocols and the extent of protection. So you all know that the mRNA vaccines have been excellent for protection against severe disease, but we really would like to have better protection against infection or mild disease, because that would help contain the pandemic, and because long-haul COVID can occur even in people with asymptomatic infection or mild disease. And then, of course, we've seen that the rapid decline in protection against infection occurs. So it's now become evident that maybe we should try other immunization protocols. So instead of only waiting 3 or 4 weeks, we maybe should wait longer between the first and the second injection. This has been proven for a number of other vaccines. But of course, for COVID, the pandemic was such a pressing emergency that they had to use a shorter immunization protocol. So what are some of the issues now? Well, mRNA vaccines are very safe, but there are some adverse events. There are tolerability issues that, while they're non-serious, they have kept people from being able to work: the fever, chills, tiredness, headache. There's also rare events of myocarditis, which, even though they haven't caused any deaths in the US to date, different regulatory agencies in different countries made different decisions about using the mRNA vaccines in younger people, particularly in males. And then there are the rare anaphylaxis and allergic reactions, which again have not caused deaths, but which are higher compared to other vaccines. And it's thought, although not definitive yet, that this is due to the polyethylene glycol that is in the formulation for the vaccines, even though polyethylene glycol is in many, many compounds such as toothpaste and laxatives. And again, comparing this with the DNA, the DNA is really much simpler because it's simply the plasmid in a saline solution. So the questions about these adverse effects are, for future use, how acceptable will the systemic symptoms be for a non-pandemic disease? So people were willing to deal with the adverse effects because the pandemic was so serious, but maybe if it's a different disease, they may just say that they will skip the vaccine. So I think that we need to work on trying to better delineate the adverse events, because maybe they are specific to a certain mRNA or certain lipid nanoparticles, or maybe they're actually specific for a particular antigen. So I think as we find out more about this, we can decrease the adverse effects. So in terms of future applications, one of the big questions is, do prior results predict the future? Well, before COVID came along, there were about 8 different entities in clinical trials of mRNA vaccines, and they gave immunogenicity in humans, but it just wasn't really very exciting, and it certainly didn't predict how wildly successful the COVID mRNA vaccines would be. So it may be that, in fact, the SARS spike antigen really is in some ways an ideal antigen. But we also have benefited in terms of the clinical protection that the SARS-CoV-2 mutation rate actually is slower than for HIV or influenza. So we still have a number of knowledge gaps, and the efficacy, therefore, for any vaccine and disease will be unique to that combination. So we have to understand the pathogen and its protective immunity. We also need to remember that results with one disease don't always translate to other diseases. So we have the example of Merck's COVID-19 vaccine candidate failing, even though it was the technology that they used for a very successful single-dose vaccine for Ebola. And of course, it'd be lovely if we could make mRNA or DNA vaccines that are single dose. I just want to mention one potential future use, and that is that the nucleic acid vaccines may be part of heterologous prime boost vaccines, which have become more acceptable because of the heterologous vaccines that have both been made for Ebola, COVID, and then the trials for phase 3 of HIV. And so before it was thought to be complicated, but in fact now they may be much more acceptable, partly because of the work done with COVID, where people got the adeno priming, and then for safety reasons, people then got an mRNA boost. Well, I'd like to just close by talking about global health and One Health. So we know that as of today, so I'm filming this earlier than the actual lecture, but only 12% of people in poor countries have received 1 dose of the COVID-19 vaccines. So the mRNA vaccines are very promising because of the large efficacy and safety databases. But the question is, will they be able to be used because of their expense and because of infrastructure problems in the world? DNA vaccines, on the other hand, are very friendly for global usage and for One Health veterinary applications. However, we really need more data to show that antibody-based protection will be effective for other diseases with DNA vaccines. And lastly, the other aspect of One Health is the environment. So the healthcare industry actually accounts for almost 5% of global CO2 emissions. If the sector were a country, it would be the 5th largest emitter. So vaccines are very beneficial in that they not only save lives and protect health, but they prevent hospitalizations and medical care, and therefore are beneficial for the environment. However, if you think of the almost 11 billion COVID-19 vaccines administered to date, you see that they also have created a huge amount of medical waste that is actually dangerous because of the sharps. So it would be nice to do needleless injection. And so I'd like to close just by saying that I think nucleic acid vaccines, both DNA and mRNA, have great potential for global usage and One Health. But for One Health vaccines, we need to have more cross-silo collaborations between animal health, human health, and environmental scientists. And we need to emphasize global solutions, not just solutions for just humans or just wealthy countries. In closing, I'd like to thank you and invite you to consider sending your work to the International Society for Vaccines Congress, which will be this fall. And we're planning to have a hybrid meeting so that you can participate as well as interact and present your work, even if you aren't able to come to Quebec. Thank you very much.
So we're back here. Thank you very much, Patricia and Margaret. So those wonderful talks, and I think we have a very nice overall on the technologies, the limitations, the potentials for each one of the different platforms, not only the mRNA but also the DNA-based platform. So I'll just invite you all to submit your questions using the Q&A box. As stated, and if you have any difficulty, please let us know. So I think we can open now the discussion, and we have one first question that I think we can make for both speakers and hear a little bit on their thoughts and views. So this first question is from Lucas Parimitre, he's a medical student. And he was asking about if you can comment on advances of the mRNA vaccine technologies designs being specific for each patient, or I mean, for each patient profile, whether expression of cellular markers would be used to select the patients. For instance, if there are any strategies addressing patients with hypertension and choose the increased levels of AC2 as an example, or for the development of a new SARS-CoV-2 vaccine. So Patricia and Dr. Liu, if you want to comment on this issue.
Thank you, Martin. So designing vaccines for each patient is very difficult in a context of public health in low and middle income countries. So I think it's more fitable if we think about chronic diseases. In fact, Martin and I were working on cancer vaccines before this pandemic, and I was working on RNA vaccines in this context. So I think in this kind of vaccine, it's more usable when you study the antigens from tumor and then design each vaccine in a context of personalized medicine. But for preventable disease, it's difficult, I think. But maybe we could design for some groups trying to skip from some problems or adverse effects that we will observe in the future. I think it's my thoughts about this.
Okay, thank you, Patricia. Dr. Liu.
Yeah, I'd just like to echo that. I think that Patricia's raised a really important point that, the questioner is making an excellent point that it's very easy to, in fact, design a new mRNA or DNA vaccine. So that can be rapidly done, and you could imagine personalizing them. And in fact, that's actually how one of the companies started off, was making these libraries where they could make personalized cancer vaccines. But the problem is whether or not for a public health type of disease, could you, even for different regions, say, design a different, COVID vaccine for different variants that have arisen? That becomes more challenging because it isn't simply about designing and making the vaccine. It has to do with all of these other issues of proving that it's efficacious. You can't simply just make the new vaccine and then start using it. As we've seen, people have asked, well, why haven't they made new vaccines against the Omicron, for example, more rapidly? And that's because they still have to do testing to show immunogenicity, even if they didn't have to show protection. But I actually wasn't sure whether the questioner was asking another question specifically about spike, because of the fact that spike is what binds to the ACE2 receptor, which of course is involved in the whole angiotensin system that regulates things like blood pressure in people. And so I just, in case that was what the questioner was asking about, I just wanted to mention that the spike that is made by the mRNA and DNA vaccines is membrane bound. So it's not soluble and therefore is largely confined to the cells at the site of injection, and therefore doesn't really get into the blood vessels where it would then bind to the ACE2 receptor on endothelial cells. So that's not actually been a safety concern per se, if that's what the questioner was asking about.
Yeah, I think, well, thank you, both of you, for your thoughts. And I think you touched the point. I think the concern was something about touching the ACE2 and some systemic blockade or activation, right? So, yes. I have a new question here for now for Dr. Liu. It's from Dr. Sang. Regarding, he's asking whether you can comment about the advantage or disadvantage of mRNA vaccines in comparison to viral vector. For instance, adenoviral vectors based vaccines.
Well, I'd be happy to answer that, since that's something that I've been, involved with in terms of the viral vector development as well as the nucleic acid vaccine. So I think that if you look at advantages and disadvantages, the advantage of nucleic acid vaccines, either mRNA or DNA vaccine, compared to any viral vector, is that any viral vector, you will get immune responses against the vector. And in fact, for some of the adenovectors, people already have been infected, and so they have pre-existing immune responses. And that may mean that that you can't reuse the vector, or even that in some people, maybe the initial response isn't as good because your antibodies would bind to the vector and therefore prevent it from infecting and transducing the cells and making the antigen. The nucleic acid vaccines, of course, are much faster to make, because to make the viral vector, you still have to remake the vector and then, make sure that your whole manufacturing process works and so on. And then, of course, as we've seen, there have been some safety concerns with this clotting disorder that's been seen, even though it's been a relatively small number of people who have died from it. Unfortunately, that's a pretty serious adverse event. And so there's concern about the vectors. Both of the two adenovectors have seen this syndrome called VIT, which is a vascular syndrome that you get thrombocytopenia and thrombosis. So that's a concern. On the flip side, there may be some differences in terms of the duration of protection, that there may be slightly longer protection or differences in T cell responses between the viral vectors and the nucleic acid vaccine. So I think that we can't rule out the utility of the viral vectors, because even the immune responses may be different. And of course, one of the really big differences, at least with mRNA, is that the viral vectors are more thermostable. So of course, DNA is also very thermostable, but compared to mRNA, which of course is both expensive and it has this issue of needing to be frozen. So that's one reason that there has been a more emphasis on using viral vector vaccines compared to mRNA in regions where there's, regions where the countries are poor and they don't have the infrastructure and don't have the super cold freezers, et cetera.
Sure. Yeah. That's why some of the formulations use different serotypes, right? So they can switch from one to the other, so they can skip this response to the first one, right? Yeah. I have a quick one for Patricia as well. Patricia, still online? I think she, okay, so let's wait for her. I have another one for Dr. Liu. And that's actually a question that rose on your talk and the potential advantages of DNA-based vaccines. And they seem to be there seems to be lots of advantage for DNA. So why was mRNA so successful in this pandemics instead of DNA? What's your view regarding this?
Wow, that is such a great question, and one that has perplexed many of us who have worked for a long time. So it was also particularly surprising because the first mRNA vaccine trials that I mentioned, there had been 8 different things looked at for mRNA before COVID. And then more recently, one of the companies, the mRNA companies, just released their phase 1 data with an influenza vaccine. And it was not particularly compelling. It wasn't really much better than other vaccines. So people really were surprised at just how potent and effective the mRNA was, and certainly compared to DNA. So I think, there's a combination of issues probably. One is that mRNA is very inflammatory. And so I think that it, in fact, does stimulate a lot of non-specific immunity that probably helps make the immune responses against the spike better. But the non-specific immunity may contribute even to some short-term protection as well, or maybe even longer-term protection. There's a whole field that's emerging of looking at these immune-enhancing effects of various types of vaccines, including actually certain live vaccines like BCG and oral polio and so on. So it may be that there was just this, the other inflammatory actions of RNA, it was just the right balance of good immune responses. But on the other hand, as I said, because the other results with mRNA have not been as exciting, they've been immune responsive, but DNA vaccines in phase 1, 2, and even some more advanced studies also were pretty good. So I think that the other issue may be, it may depend on the disease itself. So just like we saw that the Merck vaccine that was the same technology for Ebola didn't work very well for COVID, maybe it just depends. And so since people had, in some regards, had just not put as much effort anymore into DNA vaccines because they were already disappointed in them, I think it was just actually perfect timing for the particular disease and for the technology of mRNA. And I think that given the successes that have been seen now with DNA for the Indian COVID vaccine and then for all the animal uses, I think it's just going to depend that maybe for particular diseases, one vaccine technology or both or multiple technologies will work. And then other diseases such as HIV are going to continue to be a challenge, although the better technologies we have and the more we understand each disease, then I think we have a better chance of making effective vaccines.
So at the end of the day, we were very lucky to have this perfect combination, a good timing, right?
I think that's absolutely right. We were lucky, but the timing was crucial because if you think of what had to happen in the essentially 30 years since Phil Feltner first published about mRNA being able to result in protein in vivo, there had to be issues of how do you stabilize mRNA? How do you manufacture it? How do you formulate it in the lipid nanoparticles? And also what was known about the spike protein, that was based upon work of RSV that the NIH group, Barney Graham's group, had done. And if we didn't know that, to know how to stabilize the protein, the spike protein in the pre-fusion, all of this would not have, probably wouldn't have worked as well.
Sure. I have one that will go for both of you, maybe. So it's what's the biggest difficulty or limitation to work with saRNA compared to mRNA, and if there is any possible risk associated with it. And following that, how this is particularly difficult, or how difficult it is to transition from bench to manufacturing, both saRNA or mRNA. So I don't know whether Patricia is online, but the question would be for both of you, if you can comment on this.
Yeah, I think just for the audience, I think Patricia was having some internet problems. So
yeah, I'm following here on private here, and but we can go ahead, Dr. Liu, please, and she can join us.
Okay, so saRNA, which is the self-amplifying RNA, you know, it just has been very promising in terms of the preclinical work, and it actually has been in the clinic for prior to SARS as well, with the rabies, for example. But there are some, I mean, it's frankly, I've been a little surprised that it hasn't, you know, come further along, because my group used to work on replicons 20 years ago using either viral replicons or using DNA. And so certainly the preclinical work has been encouraging, but there are some issues that, for example, the production of gamma interferon helps to decrease, then there's kind of a feedback to decrease then the protein expression. And so I don't know exactly what will happen in the future with it, but I do think that one of the immunologic issues that may be an advantage for saRNA and maybe for DNA itself is there's an issue that the immune system needs to have somatic hypermutation. That's how you get high affinity antibodies. And so what happens is if you have antigen persisting, but at lower amounts, but it persists for a while, that helps to drive the somatic hypermutation. And so I think that that could be an immunologic advantage, even aside from the issues of the amount of antigen produced, for mRNA as well as for DNA compared to regular mRNA. On the other hand, one of the good things that was just recently published is that the third dose of the mRNA vaccines did result in immune responses that had been broadened. There had been some concern that that might not happen, that you might get this issue of original antigenic sin that's long been studied for influenza. And so I think we just have to wait to see what the clinical trials show, but that's a really interesting issue from an immunologic standpoint, as well as from a production standpoint about the potential differences.
So you think it is really critical, and it will take, I mean, keep expressing the protein long enough to produce these hypermutations and maturation in affinity based on these modifications of the RNA. So that's what very interesting. Patricia, would you like to comment on this question and issues raised?
Yes, thank you, Martin. As I said during my presentation, are you hearing me? Because the internet
Sure. It's
not
okay.
so good here.
It's okay.
Okay, great. As I said during my presentation, we have chosen self-amplifying RNA technology to develop our own because of some advantages we could have comparing to conventional RNA, immunological, the immunological responses, we think it would be better than the conventional RNA vaccine, and I think the major advantage is to put in line more than one antigen, because we could skip things like the variants appearance for COVID or for other disease. We are not limited to one antigen. and then we could play better with immune response, either developing antibodies and T-cell responses, what in terms of immunological responses is much better to prevent disease. So that's why we have chosen the siRNA for preventive vaccines. But I think that conventional RNA has value to when we talk about biopharmaceuticals for therapeutics. So it's difficult to think that we could use self-amplifying RNA to treat, for example, autoimmune disease. So if you think in terms of RNA being used for broad other purposes, so I think we could play with both, and I think it's not difficult to prove this in clinical assays. Self-amplifying RNA has an disadvantage. It's very long, so it's more difficult to encapsulate. But I think if we could overcome this disadvantage, the way is wide open to use this technology and apply.
And Patricia, there was a question about if you can comment a little bit more on the lipids you're using.
We are using, in fact, we now have we have our own formulation, but in order to improve this, we just signed the GDA with Croda, which owns the Avanti Technologies enterprise. So we are working together, trying to do traditional lipids formulations, ionizable lipids, together with cholesterol and pegylated lipids, and DSPC with phosphatidylcholine. So very traditional, but the difficulty is to formulate a very large RNA. It's much larger than conventional RNA.
I would like to, for both of you, maybe a comment on how much we should still play with this lipids combination in terms of not only encapsulating the mRNA or whatever RNA we're using, but also on the possibility of targeting certain cell populations and possibly modulating immune function or immune activation by using different lipid combinations. How mature do you think we are in this regard, and how much shall we explore still these possibilities?
So
maybe, yeah, please.
Okay.
It's for both
Okay.
of you. So.
It's a wish for me to move forward again after this hub implementation, to see what are the possibilities with this kind of thing, just to direct RNA to disease, for example, to see if we could decorate the nanoparticle with molecules that could address the RNA directly to the cell we want to target. And maybe it would be very useful to other purposes, like we are discussing today, for gene therapy or for siRNA therapy. I think we will develop a lot in the next years in this way.
Dr. Liu?
Yeah, I think you've made an important point about other applications. But even for vaccines, I think what we need to also keep in mind is that there is some thought that the lipid nanoparticles do play an adjuvant role, or certainly can help it, in addition to whatever immune cells the targeting occurs for. So I think to retain that activity, but something I'd like to see is if there were a way to make the mRNA vaccines mucosally administered, because it's clear that if we could have had vaccines that prevented or at least decreased infection, as well as, of course, decreasing severe diseases, that would have a huge impact on the pandemic, because then we wouldn't have all these people who are immunized, they're protected from being sick, but they're still infected. And so they are still transmitting virus. And in fact, of course, they could then be, variants could be arising within these individuals as well. So that would be something I'd really like to see, is if there's some way that the formulations can somehow then be targeted for mucosal delivery, such as intranasal delivery.
Fantastic. I would like to raise two rapid points, and we're just approaching to the end. The first one will be something that Patricia just mentioned, and I think it's a big thought. How diverse can be the load of different antigens to be loaded in this platform? And let's put it in maybe many variants we're seeing in SARS-CoV, or perhaps even more antigens when we're talking about cancer vaccines, for instance. So how limited is the number of antigens we can approach in this very same vaccine platform? And following to that, and I will ask you to be brief in the response, because we have a few minutes left, only a few minutes left. But how much this platform will really be a platform based on the point Dr. Liu rose on what a DNA platform is and what an mRNA-based vaccine cannot be really considered a platform unless we have very similar things being run in each different formulation. So I'd like just final comments on that, if you can, so we can close the session.
Well, I'll just jump in here. I think that in terms of the platform, if there's really minimal change, like just say different variant, that might very well qualify as an mRNA platform. The issue is really if you have a lot longer mRNA, or if you change the lipid nanoparticle, that's really more what I was referring to. I think that, to me, it seems like it might be really easy to just take different mRNA lipid nanoparticle formulations and mix them, so that it's just like taking different colors of balls and putting them into a mixture, rather than having to make a single vaccine encompass all of your components. Although you'd obviously still have to go through the regulatory process for the different components, but that would be more akin to the pneumococcal conjugate vaccines or the papillomavirus vaccines that have different strains mixed in.
Patricia?
Yes, I think that we are just learning how to deal with RNA. I'm suspect, but I think we have a great avenue in front of us with multiple possibilities. When you ask about the number of antigens, when we talk about self-amplifying RNA, where you could put more than one antigen, the matter is the size and not the number. The size is limiting, but we can put together more than one. And what I think is the most vaccinating thing about RNA vaccines is that I saw this in one article, that the nature of RNA never changes, and the LNP you could change a little depending on what you want, but it's not so different between one and another. So we could manipulate these things if you learn and then start doing a lot of things. So I'm very optimistic about RNA use for not only for vaccines, but also for therapeutics.
Fantastic. So I would really like to thank both of you. I think it was a very interesting, informative, and provocative session. And I would like to thank ABTCel and ASGCT for organizing this again and for having me as a chair for this discussion. And we'll have a little break now. We'll be back at 15:20 Brazilian local time. So please don't be late. And Dr. Liu and Dr. Neves, thank you again for your time and for sharing with us.
Thank you for such a great discussion.
Yes, thank you, Dr. Liu. Thank you, Martin, for your invitation.
So good afternoon, all. I'm Fernanda Cruz. I'm an adjunct professor at the Federal University of Rio de Janeiro, Brazil. So I want to thank ABTCel and also ASGCT for the invitation to chair this session. So we're going to start the second part of this meeting with our session that is entitled In Vivo Gene Editing and Disease Modeling. We're going to start with Dr. Adriana Bastos Carvalho, who is an associate professor at the Federal University of Rio de Janeiro. Dr. Adriana will be speaking today about disease modeling and gene editing in human iPS-derived cardiomyocytes. Then we're going to move to the talk of Dr. Liron Walsh, who is the head of development for in vivo programs at Intellia Therapeutics. He will be discussing Intellia's approach to in vivo gene editing and the interim clinical results from the ongoing phase 1 trial for the ATTR amyloidosis. Then we're going to end the session with some time of discussion. So I invite the audience members to engage with us in the discussion. So please feel comfortable and free to write down your questions on the question box.
Good afternoon, everyone. First, I'd like to thank the Brazilian Association of Cell and Gene Therapy and also the American Society of Gene and Cell Therapy for the invitation to speak at this webinar. Today, I'm going to show a little bit of my work using disease modeling and gene editing in human induced pluripotent stem cell derived cardiomyocytes. So here in our lab, we've worked with long QT syndrome, which is characterized by a prolongation of the QT interval in the electrocardiogram. And this is caused by a prolongation of the action potential in the heart, which can lead to severe arrhythmias and also sudden cardiac death. And the majority of cases are caused by pathogenic variants in the KCNQ1 gene, which expresses a potassium channel that is responsible for this phase of the cardiac action potential. So in this work, we have a case of a female patient that was 15 years old when she experienced a near drowning accident with cardiac arrest, which was reverted. And upon investigation, we found a prolonged QT interval of 516 milliseconds. And by Sanger sequencing, we found a KCNQ1 variant, which was an exchange of T for C at position 1763 of the coding DNA, which led to a protein change of I to T in position 588. And also, we investigated a first degree relative, but this first degree relative from which the proband was a descendant did not have an altered QT interval, but she also carried the same variant in KCNQ1. And so we were interested as to why this patient was expressing a phenotype of this caused by this variant and this patient was not, which is very common in long QT syndrome, and it's called incomplete penetrance. And there are several mechanisms that are that may be responsible for this incomplete penetrance in long QT syndrome, such as variants in modifier genes, levels of gene expression, and also environmental factors. And in our lab, we're most interested in the genetic aspects of incomplete penetrance, which we'll investigate in this talk. So we generated induced pluripotent stem cells using peripheral blood from both the proband and the first-degree relative. And this was conducted using Sendai virus expressing the Yamanaka factors. So we generated the iPS cells from both the proband, which I am calling long QT syndrome 1 from now on, and from the first-degree relative, which did not express a phenotype, so I will be calling it a carrier. And these iPS cells had a normal carrier type. They expressed high levels of several pluripotency factors, such as OCT34, SOX2, NANOG, Tri181, Tri160, and by both flow cytometry and RT-PCR. We did continued characterization of the cell lines, and we conducted a 3 germ layer differentiation in these cells to demonstrate that they had the ability to differentiate into the 3 embryonic germ layers. And we found both expression of nestin indicating ectoderm differentiation, brachyury indicating mesoderm differentiation, and HNF4A indicating endoderm differentiation in both cell lines. So after we concluded the characterization of the iPS cell lines, we moved into cardiac differentiation and disease modeling. So we used the Palachek protocol to differentiate these cells into cardiomyocytes. As you can see, they start spontaneously contracting in culture. We also checked efficiency of differentiation, which was above 90%, using flow cytometry of troponin T, cardiac troponin T in these cells. And then we went on to measure action potential duration for both the cell lines. And as you can see, the LQTS1 line is significantly, has a significantly longer action potential duration when compared to the carrier, which is expected. And this demonstrates that the iPSC model reproduced incomplete penetrance that we had found in the patients. And so we went on to try to do some gene editing to confirm variant pathogenicity. So when we started working, and here I'm going to talk a bit about the technical aspects of this process for anyone in Brazil who's trying to implement this in their lab. We started working with plasmids expressing Cas9 and the guide RNA, which also had a selection cassette. We had acquired these plasmids from Adgene, and also using the donor molecule, and we would combine this using a neotransfection system to try and to get editing in the iPS cell lines. And we tried this for at least 6 or 7 batches of, of the protocol, and we couldn't get good results. The cells would either die or they would not be selected when we started using the antibiotics. And after a lot of trial and error and unsuccessful tryouts, we decided to change the system, and we started using the TrueCut Cas9 protein. The protein is directly administered to the cells instead of being expressed by the plasmid, and also with together with the guide RNA and the donor sequence, and these were transfected into the cells. And so after we changed this protocol, we started trying to remove KCNQ1 variant from the proband to demonstrate that if the variant was removed, we would get a reduction in the duration of the action potentials. So after we transfected the cells with the Cas9 protein and the oligos, we did a limited dilution plating of the cells, so we would get clones from each of the cells that might be or not edited, as this is not a very efficient process. And then after we got these small clones, these clones were picked. We picked 50 colonies from the plate after we did the gene editing protocol. Of these 50 colonies, we sequenced 20 clones, and we got from these 20 clones, 3 clones that were corrected. And this is the demonstration of the correction in the patient in the LQT1 cell line from the proband. in which we used to have the variant, a C in exchange of a T, and now we have a T in both alleles of this cell line. So we got the correction in this line. So after that, we again submitted the cells after they were corrected to cardiac differentiation. And we went again to measure the action potential duration of these cells. And what we saw is that comparing the same cell line before correction and after correction, we see a significant reduction in action potential duration, which demonstrates that this variant, this KCNQ1 variant, has a role in a pathogenic role in prolonging action potential in these cells. So after we demonstrated that KCNQ1 variant was pathogenic, we went to investigate the mechanisms of incomplete penetrance, and we were especially interested in variants in other genes that might modify the phenotype. And so what we did is that we submitted both the LQTS1 and the carrier iPS cells to exome sequencing, looking for other variants that were different between the two lines. In these lines, we found 33,000 variants that were the same between the two lines. 10,000 were exclusive of the proband, and 11,000 were exclusive of the carrier. And we were looking for differences between these two cell lines that might explain the difference in the phenotype. So we had a total of 22,000 variants that were different between the two cell lines, but we selected 372 candidate genes, which was based on genes that are already known to cause channelopathies, because these genes probably would also alter QT interval, and also genes that have been related to the QT interval by previous genome-wide association studies. And out of these 372 candidate genes and candidate variants that were different between the proband and the carrier, we found one variant that was present in the SCN5A gene, which codifies the cardiac sodium channel. And this is an exchange of an A or G at position 1673 of the coding DNA, which leads to a protein change of an H for an R at 558 position. And as you can see, this variant is present in the LQTS1 line and is absent in the carrier line. And we had evidence from the literature that variants, polymorphisms, or variants in these genes, SCN5A and others, could impact QT interval and also arrhythmia risk in these patients. And this had been described previously. And so we went to investigate if this might be the cause of the difference in phenotype between the two cell lines. So we inserted the SCN5A variant in the carrier by gene editing using the same protocol that we did before with the Cas9 protein. And what we saw, we submitted again the cell lines to limiting dilution. We picked 20 colonies and sequenced the 20 clones, and we got one clone corrected in this case. And here's the result. We are trying to insert the variant in the carrier line, and here you can see that both alleles show a G, demonstrating that the line was corrected. The only observation is that this was in homozygosis. It's very hard to get gene editing in heterozygosis because both alleles will tend to be corrected, but in the proband, the LQTS1 line was a heterozygote, and but we are modeling, and we are still working on getting new editing in these cells. But from this line, what we saw after submitting the cells to cardiac differentiation is that there was a significant prolongation in action potential duration after we inserted the SCN5A variant in the carrier cell line. So as conclusions, iPSCs can be used to model incomplete penetrance and study modifier genes. The KCNQ1 I5AT correction reduced action potential duration in our lines, while the SCN5A insertion increased action potential duration in a line that was a carrier of the KCNQ1 variant. The LQTS1 phenotype is modulated by a SNP in the cardiac sodium channel, and also that at least in our hands, using the Cas9 protein instead of plasmids led to more efficient and easier obtention of gene editing in our cells. And I thank you very much for your attention. I would also like to acknowledge the work of the people that conducted these experiments, especially Hayana, which is a postdoc in our lab. If anyone wants technical information on our protocols, please feel free to contact me and I'll put you in contact with Hayana. And also acknowledge the funding agencies that fund our work. Thank you very much, and I'll be happy to take questions.
Good afternoon, and thank you for inviting me to speak at this session. On behalf of Intellia, the patients and their families in our clinical trials, I'm excited to present our approach to in vivo genome editing and review the interim results from our most advanced program, NTLA-2001, which is currently being studied in a phase 1 trial for the treatment of ATTR amyloidosis. As for my disclosures, I am an employee of Intellia and currently lead the in vivo development efforts. NTLA-2001 has not been approved by any health authority. I'd like to start off by providing an overview of Intellia's approach to genome editing. Treating a broad range of severe diseases will require multiple approaches. For genetic diseases, we are leveraging our proprietary lipid nanoparticle delivery system to selectively inactivate disease-causing genes or precisely insert genes to produce desired proteins. Our ex vivo approach leverages our differentiated cell engineering platform to produce a homogeneous, robust cell product to fight cancer or address autoimmune diseases. My focus today will be on our in vivo approach, utilizing our lead program NTLA-2001 as an exemplar. Before we move on to the interim results, I want to highlight the potential advantages of Intellia's in vivo approach. First, this approach allows us to bring forth potentially curative therapies for genetic diseases where there is an unmet need following a single dose. This could have tremendous advantages for both patients as well as the broader healthcare system. Next, systemic non-viral delivery of CRISPR-Cas9 using our LNP technology provides transient expression of both the delivery vehicle as well as the CRISPR-Cas9 RNA cargo within the LNP. The transient expression confers a number of potential advantages. Next, our technology allows for permanent gain of function with targeted gene insertion when developing treatments for diseases where the patient is missing a critical protein. Let's move to slide 5, where I present the interim clinical data from the phase 1 NTLA-2001 clinical trial. Transthyretin amyloidosis is a rare, progressive, and fatal disease caused by accumulation of amyloid deposits composed of misfolded transthyretin protein. Transthyretin amyloidosis consists of 2 forms, hereditary and wild type, which cause a spectrum of clinical disease. The rate of new diagnoses is increasing, where the hereditary type is estimated to affect 50,000 individuals worldwide. It has a variable phenotypic presentation that primarily features either peripheral and autonomic polyneuropathy or cardiomyopathy. The wild type is primarily a cardiomyopathy that is estimated to affect upwards of 500,000 people. It is an increasingly recognized cause of heart failure in individuals over age 50. It is progressive and typically fatal within 3 to 10 years. It has recently been recognized that the majority of affected individuals are never diagnosed. Therapy in ATTR amyloidosis is directed to reducing the circulating amyloid-forming protein. Long-term repeated administration of gene silencing therapies, such as siRNA and antisense oligonucleotides, decrease TTR mRNA and reduce serum TTR by approximately 80%. Long-term therapy can result in stabilization of neuropathy, and greater reductions of TTR is expected to lead to better clinical outcomes, including improvements in quality of life and more pronounced regression of disease. NTLA-2001 is being studied as a potential one-time treatment to knock out the TTR gene for the treatment of ATTR amyloidosis. NTLA-2001 is an investigational CRISPR-Cas9-based in vivo gene editing therapy. It consists of a single guide RNA molecule that targets the human TTR gene and a human-optimized mRNA sequence of strep pyogenes Cas9 protein, encapsulated in a lipid nanoparticle. This cartoon summarizes the mechanism of action of NTLA-2001. Following intravenous administration, the lipid nanoparticles are transported directly to the liver, where they are taken up via the LDL receptor on hepatocytes. After endocytosis and release into the cytoplasm, the Cas9 mRNA is translated, producing the Cas9 enzyme, which then interacts with the single guide RNA to form a CRISPR-Cas9 ribonucleoprotein complex. This complex enters the nucleus, and a 20 nucleotide sequence at the 5' end of the guide RNA binds to the patient's double helix at the complementary target site. This leads to precise knockout at the targeted TTR gene sequence, which consequently stops production of the transthyretin protein. A critical component to our therapeutic approach is evaluating the potential for off-target edits. To that end, we use complementary methods designed to initially cast a wide net to discover potential off-target sites, which then undergo robust validation. The left-hand slide of the figure highlights our discovery efforts and utilizes 3 approaches. The first utilizes cast-off finder, which leverages computational predictions to search for potential off-target sites. The second uses site-seq, which is a cell-free biochemical method that is among the most sensitive to potential off-target editing. In this method, purified genomic DNA is digested with NTLA-2001. The third method, guide-seq, is a cell-based assay that relies on the incorporation of double-stranded DNA oligonucleotide into double-strand breaks created by NTLA-2001 in living cells. The combination of all sites from these methods are accumulated to define the potential off-target landscape. All potential off-target editing loci discovered were validated for the detection of off-target indels with the use of next-generation sequencing after NTLA-2001 treatment of primary human hepatocytes. These results are presented on the next slide. Validation of potential off-target editing in primary human hepatocytes identified 7 validated loci in non-coding regions after exposure to supersaturating concentrations of NTLA-2001. No evidence of off-target editing was detected at NTLA-2001 concentrations 3-fold greater than the EC90, which is the concentration at which 90% TTR protein reduction is achieved. These data demonstrate the high specificity of NTLA-2001 for the TTR gene. Preclinical in vivo studies were conducted in non-human primates. This graph shows the mean percent reduction from baseline in serum TTR concentration against time following single intravenous doses of 1.5, 3, and 6 mg per kg on day 0 and followed for over 360 days. A control cohort received no treatment was included for comparison. Vertical lines represent standard deviations across the 3 animals in each group. 2 important observations can be made from these results. First, there was a dose-response relationship with the highest dose achieving more than 95% reduction in circulating transthyretin and lower doses demonstrating a more variable response. Second, the response was durable for at least 1 year after the single dose. Safety studies demonstrated that the adverse effects of NTLA-2001 were dose-dependent, transient, reversible, and monitorable. A dose of 3 mg per kg in non-human primates corresponds to approximately 1 mg per kg in humans. This arm in the first-in-human study with NTLA-2001 is a 2-part, open-label, multi-center study in patients with hereditary ATTR amyloidosis with polyneuropathy. Part 1 is a single ascending dose portion of the study, with a minimum of 3 patients in each of the 4 dose escalation cohorts of 0.1, 0.3, 0.7, and 1 mg per kg. Part 2 will consist of administration of the dose selected from Part 1 to a dose expansion cohort in at least 8 subjects. The primary objectives of this first-in-human study are to evaluate safety, tolerability, PK, and PD, the latter by serial measurements of serum TTR. I will report data on all 15 patients enrolled in the 4 dose escalation cohorts included in Part 1. This table summarizes the demographics of the 15 study participants. Median age was 55, with range of 19 to 70 years. 9 men and 6 women were enrolled. Most subjects self-reported as Caucasian. Median weight was 83 kilograms with a range of 59 to 111 kilograms. 7 different mutations were represented in these 15 patients. Most patients had early disease with mild neuropathy and minimal or no cardiomyopathy. 2 subjects had peripheral neuropathy with a disability score of 2, and 1 subject had cardiomyopathy with New York Heart Association Class 2. 7 of the 15 subjects had elevated proBNP measurements at baseline. This slide summarizes the safety data from the study. NTLA-2001 was generally well tolerated across all dose levels. All adverse events, regardless of a relationship to treatment, are presented. The most frequent adverse events were headache, infusion-related reactions, back pain, rash, and nausea. In the majority of patients, the maximal severity of adverse events through the entire observation period was mild. Infusion-related reactions were typical for those observed with LNP infusions, characterized by back pain, headache, or fever. These were all graded as mild and resolved within 24 hours without clinical sequelae. All patients completed their infusions, receiving their full intended doses, and remain on study. There was a single serious adverse event of vomiting in a patient with a pre-existing history of autonomic neuropathy and a prior hospitalization for gastroparesis. This dose-limiting toxicity, considered possibly related to study drug, required expansion of the 1 mg per kg cohort to a total of 6 subjects. The cohort completed enrollment with no additional dose-limiting toxicities. No clinically significant laboratory findings were observed. Transient grade 1 liver enzymes were observed, which resolved shortly following infusion and were not recorded as adverse events. The maximally tolerated dose was not reached. The next 4 slides summarize the pharmacodynamic effect from Part 1 of this first-in-human study. The first figure depicts the mean TTR reduction from baseline at 28 days post-treatment for each of the 4 cohorts. Doses of 0.3 mg per kg and above achieved mean reductions of greater than 80%, and the highest dose of 1 mg per kg achieved a mean reduction of 93%. There was a dose-response relationship, but increasing the dose from 0.1 to 0.3 mg per kg resulted in the largest incremental decline in serum TTR than increasing from 0.3 to 1 mg per kg. I should note that 0.1 mg per kg was the first ever in-human systemic administration of a lipid nanoparticle CRISPR-Cas9 gene editing therapeutic. The purpose of this dose cohort was to evaluate safety. It was encouraging that not only was the agent safely administered, but all 3 subjects had measurable reductions in serum TTR. We intend to offer retreatment to these subjects with a higher dose level of NTLA-2001 and report on the results in the future. Closer inspection of the data in the 1 mg/kg cohort revealed 4 of the 6 subjects had reductions greater than 93%. All subjects had reductions in serum TTR at day 28 that were within 10% of the mean and greater than 80%. These findings on magnitude and consistency, as well as the findings at 0.3 and 0.7 mg/kg, suggest this dose has neared a saturable effect. This figure depicts the mean percent reduction from baseline of serum TTR plotted at 7, 14, 28, and 56 days post-infusion for each of the 4 dose cohorts. Similar to the non-human primate data shown earlier, dose-dependent reductions in serum TTR were observed, with nadir reached at approximately 28 days. This figure depicts the durability of effect of NTLA-2001. In the bar graph, each pair of bars depicts the mean percent TTR reduction for a dose cohort, with hashed bars showing the mean reductions at day 28 and solid bars the mean reductions for the latest measurement for each patient. The duration of follow-up ranges from 2 months for the most recent cohort up to 12 months for the first cohort. The mean reduction in TTR observed at day 28 was sustained through the latest available time points. For example, in the 1 mg/kg dose cohort, the mean reduction was 93% both at day 28 and the last available follow-up. In conclusion, a single intravenous administration of NTLA-2001 in patients with hereditary ATTR amyloidosis and polyneuropathy achieved rapid and profound dose-dependent reductions in serum TTR protein concentration, with 0.3, 0.7, and 1 mg/kg cohorts achieving a mean decrease from baseline of 87%, 86%, and 93% respectively. Reductions in serum TTR were maintained through the last available follow-up measurement. NTLA-2001 was generally well tolerated, with nearly all adverse events being of mild severity. The maximum tolerated dose was not reached. Based on the safety, activity, and pharmacokinetic profile of NTLA-2001 observed in Part 1, a fixed dose of 80 milligrams has been selected for evaluation in Part 2 of this trial. 80 milligrams is expected to result in consistent and deep reductions in TTR comparable to 1 mg/kg. These data confirm and extend the early findings reported last year on this pioneering trial, demonstrating the promise of CRISPR-based in vivo gene editing in humans. Contributions of numerous individuals made this project possible. Foremost, we thank the patients who participated in this study and their family and loved ones. I would also like to thank our investigators and their research staff. Thank you for your attention.
So I'm going to invite Dr. Adriana and also Dr. Walsh to the discussion now. We have been seeing on the 2 talks that gene editing can be used for proof of concept studies, but also for treatment studies. And I want to start this discussion with the provocative questions for both of you. So when we are talking about gene editing, should we ask whether we'll be able to use this technology in the future before we start asking how does this technology really work? So I want to ask both of you, how do you feel about the regulation aspects in both countries?
So I'm happy to start, and thank you again for inviting me. What's really amazing about the technology is the ability to get to precisely deliver therapy to the locus of the disease, so to speak, whether that's in the liver and perhaps in the future elsewhere. I think that the regulations with respect to studying these are consistent with a responsible and ethical clinical development for these therapies. And so in Europe, as well as the FDA, I don't know about Brazil, but have really strict guidelines about ensuring that the preclinical package, supports moving into patients, as well as that there's long-term follow-up up to 15 years at least to ensure that these patients are doing well over the long term.
So Adriana,
thank you. Yeah, thank you, Fernanda, for the question, and thanks again for inviting me. In line to Liron's comment, Brazil has also a very strong regulatory agency which revises preclinical data before allowing use of this new technologies in patients. But I don't believe we're close to being ready to move into clinical use of gene editing in Brazil, because we're still starting with the preclinical work. And of course, as Fernanda commented, off-target effects of gene editing are something to worry about. And I'm actually working with ex vivo in vitro gene editing, but I'm here together with Liron in this talk. But we are also worried from a scientific standpoint, and we are going to look into off-target effects by exome sequencing in our lines. And this is something we have to always check for in preclinical and clinical work. So it's a much valid point.
I'm going to bring Dr. Sen's question. So he's asking you, Adriana, so if you can comment about your study, how can your study be translated into clinical studies? And considering that the heart is composed of several cell types, can you comment on that?
Well, as I said, I'm still working with the in vitro part, but there are many genes that are exclusively expressed in the heart, but the ion channels are not one of them. There are ion channels in other cells, neurons, for instance, several other organs. So we have to be, worried of these potential off-target effects. But I believe, moving forward, as shown with the TTR gene, we also have a lot of options for cardiac inherited diseases, which are much, much more common than people think. For instance, hypertrophic cardiomyopathy is an incidence of 1 to 500 cases worldwide, and so we are excited to look into how this will develop in the near future for these diseases.
So still complementing the previous question, Adriana. So can we expect that this single nucleotide polymorphism to play roles in other tissues as well? So should we investigate the possibility of gene editing in other cell types?
Well, that's a great question. So incomplete penetrance is very, very common in cardiovascular inherited diseases, especially lung kidney syndrome. It's very, very variable. And so you may see here in Brazil, we have large families with a lot of genotype positive individuals that do not develop phenotype. And so that's the reason why we're interested in the SNPs. But, there has to be, if in the example I showed in my talk, it's LQ2S type 1 phenotype, and we know that the gene that causes the phenotype is KCNQ1. So I don't believe we're going to have to edit the SNPs. I believe we'll edit the causative variant, and this should lead to a resolution of the phenotype. But the oligogenic nature of previous monogenic diseases will certainly be an issue for gene editing in the case of diseases that are strongly oligogenic instead of monogenic. And literature has been growing a lot in this aspect, showing that several diseases that we believe to be monogenic are actually oligogenic. And this is something that we have also to be careful.
Thank you, Adriana. So I'm going to move to Dr. Walsh. Have you seen any difference of efficacy between young versus old patients? As efficacy depends on the transfection efficiency, it seems to have some variation among different ages. So can you comment?
Yeah, it's a good question and something we thought about, but when we studied this in patients, we honestly haven't seen a difference in terms of the reduction in TTR, particularly at higher doses. So where we get, approximately 90+% reduction in TTR, and there doesn't appear to be therefore any variability across age groups.
So two more questions for you, Dr. Walsh. So is there any additional underlying conditions that you can anticipate as a potential limitation for this in vivo delivery?
Yeah, it's another good question. As we continue to develop 2001 and other therapies, we'll have to understand what the safety profile looks like of those various therapies in order to better assess who might be the right patients for treatment. At the outset, as you would expect, we're choosing patients that have a healthier milieu, although they still have the underlying disorder. As we gain confidence in the safety and efficacy profile, we'll likely be able to expand that, and we'll understand further where the limitations are. The toxicity is primarily acute in nature, so it's that delivery of the LNP-based CRISPR-Cas9 therapy, which is transient. So any long-term toxicity will not be related to any of the LNP-related activity.
Still on this topic, I'm gonna move back to Adriana because Dr. Walsh has shown really impressive results on the reduction of the TTR protein with different concentrations. And in your in vitro studies, Adriana, you have shown a little bit of a small efficiency on the transduction of those cells. So what is your group thinking about it? So is this a protocol thing that you have to manage, or is this something special to the cells you're working with, or is this a concentration curve that we should proceed? So what are you thinking about it?
Well, I mean, the strategies are quite different. We still have, because in our design, we're actually isolating a single cell that got corrected and then doing functional experiments after that. So we're going to see, 100% corrected cells and what that looks like from a functional standpoint. We haven't done protein levels yet. We're still going to do our Western blots in that aspect, but we have done the most relevant functional assay for a cardiomyocyte, which is the action potential. And so when we get 100% corrected lines, because we have that coming from a single clone, we do see a difference, a relevant difference in the functional aspects. But as you were mentioning, we don't get very high efficiency. We didn't get before when we were using the plasmids. It got extremely improved with the protein, but it's not, not even close to getting all the cell lines corrected. The point is, we don't really know in vivo how many cells do get corrected, and we don't know what's the relationship, if we need a linear relationship of corrected cells to get such an increase in protein, wild type protein expression. So it may be that they're correcting 10% of the cells and getting a lot of protein, wild type protein expressed. And I don't really know if we already have enough evidence to know how many cells we need to get corrected to have such an increase or a decrease in vivo in these proteins. So it's something that remains to be determined. I don't know if Dr. Walsh wants to comment on it. But with regards to the question, if we are studying different concentrations, we haven't been working much on titration of the protocol. We got it working, we got the clones, and we started looking at them functionally, but it's certainly something that should be done in the future.
I don't have more to add to that other than that this is really hard work. So science takes time.
Yes. So Dr. Walsh, we have another question for you here. So are liver biopsies plans to in loco evaluating gene editing and potential of target outcomes along with potential clone-based expansions?
Yeah, that's a good question. At this time, we aren't planning on performing liver biopsies. It's an invasive procedure, obviously, and has its own potential complications. Our off-target assessment is extremely robust, so we're quite confident that NTLA-2001 is highly specific for the TTR gene, making the likelihood of off targets, extremely small.
Your group have tested the IV injection of the components, right, for the gene addition. So what is your group thinking? Which are the cells that are being edited after the IV injection?
Yeah, so it's predominantly the hepatocytes, because the uptake is predominantly in the liver. It's in the hepatocytes themselves, which is exactly where we want it to go. And the LNP technology allows us to hone into those hepatocytes.
Fernando, if I may comment, I think all the gene editing clinical trials that have started so far are targeting the liver. I don't know if any other organs being targeted yet. I don't know if the heart being targeted yet, but most of them are targeting the liver. Yeah. Right, Dr. Walsh?
That's correct, unless it's ex vivo, but they're all targeting the liver that's in vivo. Even the gene therapies are essentially liver-based targets. So
So are you planning on extending the time of that your patients are going to be, that this phase 1 trial patients will be accompanied? Or I have seen that you have for 6 months for the higher doses, but
we'll be following them up for at least a period of likely 15 years based on the regulations, and to ensure that we continue to have favorable safety and activity.
And are you planning on doing biopsies at least in primates or in experimental animals?
So we've done biopsies in rodents, and we've seen 80% or so gene editing, I believe, but I'd have to check on that.
Yeah, so I think we have finished our questions here, and I'm gonna thank Adriana and Dr. Liron for the awesome talks and for being able to discuss with us. So I'm gonna thank again the ABTCel and also the ASGCT for the opportunity.
Hi everyone, we are back. I am Guilherme Baldo, and I will be moderating this last session from today's meeting. In this session, we have 2 speakers. The first one is Dr. Ricardo Weinlich, who is a scientist at Einstein Hospital in São Paulo, here in Brazil. He will be followed by Dr. Matthew Porteous, who is a professor of definitive and curative medicine at Stanford University School of Medicine. They will be both sharing with us their experience with ex vivo gene editing programs. So thank you, Dr. Weinlich and Dr. Porteous, for accepting our invitation. After the 2 talks, we'll have our Q&A session. So please type in your questions for our speakers so we can have a discussion after the talks. Once again, thank you for inviting me, and thank you, Dr. Weinlich and Dr. Porteous, for being here with us. So I believe we can start our first talk from this last session.
So good afternoon. My name is Ricardo. I'd like to thank Guilherme and all the organizers for the opportunity to be here with you and share some of our results that we are very excited for. So today I'll present data produced by this team, the Einstein Enterprise in Gene Therapy. This photo is not updated due to COVID restrictions, but all the names are down here. And we also would like to thank the partners, Dr. Wanderson Rocha from University of São Paulo, and Dr. Nelson Hammerschlag and Dr. José Mauro Kuttner from our institution as well. This project is supported by the Ministry of Health through a very nice program called ProAres SUS. and I have no conflict of interest to disclosure for these projects. So one of the projects that are most advanced, and I'd like to share with you the data, is the sickle cell disease project. The main aim of the project is to cure sickle cell anemia patients by correcting the causative mutation that happens in the beta-globin gene. And we are going to do that by autologous infusing CD34 stem cells that we correct the mutation in vitro using the CRISPR-Cas9 system. Just to briefly mention the disease here. So the disease is caused by point mutation in most cases that changes an amino acid in the beta-globin gene, right? So when this happens, the protein, the beta-globin that it's dimerized with alpha-globin to build the beta-globin, it starts to polymerize into fibers and change the format of the red blood cell into this sickle cell format here, right? And this is not only bad for the oxygen transportation, but also alters the dynamics of molecular expression in their surface, making them more adhesive to the endothelia and activating the endothelia, causing these vaso-occlusions here. And this causes a lot of symptoms, both chronic symptoms and acute complications that are quite bad for the patient. These patients suffer from all these issues here, and they feel a lot of pain, chronic pain and acute pain. And so it's a patient that has to visit the doctor and the hospital frequently. This causes a lot of morbidity to the patients and also increases the mortality. So the life expectancy of these patients is 20-25 years less than the average population. And although it's a rare disease, it's the most common genetic disease in some of the countries, right? Like in Brazil. So in Brazil, we have around 40,000 patients with sickle cell anemia that could benefit from curative treatment. And the disease is not only bad for the patient, but it's also associated with a high hospital cost. So for example, in the US, it was estimated that per year, this disease costs more than $1 billion. And as you can see, the distribution in this map of the frequencies of the estimated frequencies of birth rate with sickle cell anemia, this disease is most prevalent in countries that are underdeveloped, putting even more pressure into their health system and their health insurance system. So until now, this disease is managed using pain medications, RBC transfusion or antibiotics, and is treated with hydroxyurea that induces fetal hemoglobin, which is a surrogate for the adult hemoglobin. But the only curative treatment for the disease is the allogeneic stem cell transplantation, especially from identical siblings. And this works pretty well when you have the compatible donor. It's curative. So it's a hint that we can really cure this disease. But as you know, to find a compatible donor is quite difficult. And the stem cell, allogeneic stem cell transplantation also carries a lot of complications after the transplant, as I'm showing you here. So if we could use the cells from the same patient and correct this mutation in vitro and using the same cells, that will be a great advance in terms of curative opportunities for these patients. So that's why we started working on a protocol that is represented here. It's a simplified version of it, of course, but what we would do is to mobilize cells from this patient. Then we would collect the peripheral blood cells and purify the CD34 stem cells from these patients. Then we take these cells to the lab. We edit the cells with the CRISPR-Cas system. Then we do all the control of quality. And then once the cells are ready to go, we call back these patients. We prepare these patients for the infusion, and we infuse the cells into these patients. And then we have to follow this patient, but in principle, this would be a curative protocol. And why the CD34 cells, right? So the cells that are affected by this disease is the red blood cells here, because they express the hemoglobin, the beta globin gene, right? But these cells are short-lived. So if you correct directly these cells, you wouldn't have a long-term curative treatment. So we go all the way back here, and we try to correct these cells here, the long-term stem cells. And all these cells, the progenitor cells and the stem cells, they all express the CD34 marker. So the idea here is to collect the cells, edit the cells, and give them back to the patients. As you can see here as well, the long-term stem cells, they have different markers from the progenitors. So we are also thinking in separating specifically the long-term stem cells and only editing these cells. But for now, I'll show the results with the bulk CD34 stem cells and progenitors that are showed here in this figure. So the lab protocol here is to collect the cells from different sources. We use cells from health donors and from exchange transfusion from sickle cell disease patients. And we take the cells, the CD34, and we stimulate them with a cytokine cocktail in order for them to start proliferating. And then after a couple of days, we do the editing. And we do that by electroporation of the Cas9 pre-complexed with the guide. And together with that, we give the donor DNA through AAV6 vector. So after another couple of days, we will evaluate, and we have evaluated, the frequencies of allele correction, the immunophenotyping, the viability of the cells, but also what is important is to show that the cells are still functional, either in vitro or in vivo models. So here's the first result that I'd like to show you here, is the percentage of corrected alleles, right? So here we have two groups of cells, the cells from health donors and from patients. So here in the health donors, we can assess the target correction by silent mutations around the specific mutation from the HBB locus that's causative for the SCD disease. So here you can see that it doesn't matter if we use a lower MOI or a higher MOI, we can get good numbers of target alleles that are corrected here, both in lab conditions or in GMP conditions as well. And for patients that we use cells from exchange transfusion, we also get quite good numbers of corrected alleles. You can see here that we don't affect the viability of the cells compared to the mock situation for the health cells or the SCA patients as well. Again, I was telling you, it's not only vital to show that you target correctly the alleles and you were able to get high frequencies of cells corrected, but you also need to show that these cells are functional. So I'll show you here two in vitro assays to show that. The first one is the erythroid differentiation, when you force the cells to differentiate to the red blood series, right? So you can follow the markers, the cell surface markers, to show that these cells that were added are differentiating normally. And here you show for health donors and patients that it doesn't show any different from the mock condition, that the cells do differentiate. And most important, the cells that do differentiate, they are the cells that are bearing the corrected alleles. As you can see here, we don't lose a percentage of corrected alleles in these populations, both for the health donors and the patients in either MOIs, right? So the cells that were touched by the gene editing, they do differentiate it without any issue. And if you look here, not only they bear the corrected allele, but they start expressing the healthy beta globin. So if you compare here with the mock situation, you have mostly the beta S, which is the sickle allele, but then with the treatment here, you can reduce the sickle allele, and you start to express the functional allele, the healthy allele, showing these cells are expressing the correct beta globin, and this is it's quite a good characteristic of this cell. The other functional assay that we are showing here is a stromal cell supported CFA, and the aim here is to show that the cells don't lose pluripotency, right? So we single cell sort the cells, and we observe if each of the cells can generate a colony, and we observe which type of colony is made here, right? So as you can see here, not the numbers nor the type of colonies that are generated are different from our treatment here from the mock, so they're comparable here. So the cells maintain their potency, and these cells that were forming colonies, they were cells that were genetically corrected, as you can see here. These are the genotyping of the colonies, and you can see here the AGR, which means that the correct allele is present in both alleles or in just one allele here, and you can see that the numbers are pretty high. The next step is to show that these cells can survive in an animal model for a longer period of time than in our in vitro experiments. So again, you inject the corrected cells into these animals, and you check for engraftment, type of cells that are derived from this engraftment, and if they possess the correct alleles. And you can see here, they do engraft 100% animals of this experiment here, and we repeated this experiment a series of times. And this, so they do engraft, and they do derive B cells and T cells and a little bit of stem cells as well, with no different from the mock situation here, showing that the cells can stand a long time in these animal models. And once we sacrifice these animals and we get the cells out of the animals, you can see here that they still present the correct alleles in a frequency that is compatible with the input material that we put in these animals. So that's quite good. We are very satisfied. We are now trying to move this to a larger scale to start the preclinical studies for the submission for our FDA-like agency. But we were also concerned by possible off-targets or undesired effects. So what we did, apart from looking for the off-targets by in silico and in vitro analysis, we also ran this sequencing platform panel here that checks for most of the oncogenes that are known to be important for a series of genes with a high coverage here of the genes that are involved in these diseases. And we did the pre-correction and post-correction, and we didn't see any alterations specifically driven by the genetic editing in the cells, which is quite comforting. On the other hand, we look for larger deletions on the proper locus of the HBB locus. And as you can see here, we have a fairly high frequency of large deletions in these cells that if you check later on after the differentiation assay, this tends to diminish and vanish in some cases, but it still is something that we want to investigate further to make sure that the cells here don't represent any risk for the patient once they are transplanted into them. So just to finish with some different aspects of our initiative here, and that's why it's so good to have a great team working with us, is that as we were trying to diminish the MOI that we were using to correct these cells, there's a limit that you go under that is, it's no good, right? You don't get enough alleles that were corrected. But looking in other situations, one of our specialists found that adding this compound A that's used in the clinics in a completely different aspect, it increases the efficiency of the correction, and we can even lower down to 4 times less virus, and this is comparable to what we were using and we were seeing before. This compound doesn't affect the in vitro assays, and we can see that the cells, they do make the colonies and they do differentiate. So it's quite impressive with what this compound A can do to increase the efficiency in gene correction. So we are investing in maybe changing our protocol, current protocol, to add this compound to it. And again, we are also trying different approaches. Here, just sharing this slide with you. This is my final slide to show you that instead of using the AAV6 as a donor DNA, here we use the ASGCT approach. So we designed 5 different ASGCTs, and here is the ASGCT that were previously published. And as you can see here, our ASGCTs, both for gene correction and viability, are as good as or even better than the published version here. So we are quite excited to use also this approach in future clinical protocols. And this method here is interesting because it's simpler and cheaper than the AAV6 approach. So we are trying to be very creative here to make these protocols less expensive, more affordable, as we are aiming specifically to treat the population in Brazil through the public system. So with that, I bring again the team that do most of this work here with our partners, and I'd like to thank your attention. And I'll leave here my email if you want to contact me, and also our Instagram handle where we do some scientific outreach. Again, thanks, Guilherme, and I'll give the word back to you. Thank you.
Hello, my name's Matt Porteous, and I'm a professor of pediatrics and in definitive and curative medicine here at Stanford. And I'm excited to be able to follow Ricardo in his presentation of their ex vivo genome editing approach for sickle cell disease, and I'll describe sort of our parallel work. And again, I'm excited to be part of this joint workshop and symposium today. Fortunately, genome editing has become a very hot field, and I've been lucky enough to be involved in several different biotech ventures, the most relevant of which today is Graphite Bio, who is now sponsoring the clinical trial using gene correction for sickle cell disease in the United States. I'm going to start at the very high, broad view and highlight the optimism that many of us feel about the beginning of a new era in using what people are calling living drugs to treat patients for which prior modalities have been ineffective, which is not to say that things like sanitation, anesthesia, vaccines, small molecules, or biologics have been unimportant. Of course, they have transformed the lives of billions of people around the world currently and in history. Nonetheless, there remains an important group of patients whose diseases are still not well treated by these past modalities. And to give a visual example of what we mean by a living drug is shown here, in which we use a genome editing process that I will describe in more detail, and Ricardo just described as well, in which we integrate a luciferase gene into the beta globin locus in mouse, in human CD34 positive cells. And then we transplant these cells into immunodeficient mice and visualize them over time. And what you can see is that at day 0, when the cells are injected, there's sort of this background light emission throughout the body. But within 3 days, the light emission localizes to the site of the injection in the left femur. And over time, these cells migrate from the left femur to the right femur, and increasingly over months, they migrate to essentially all of the blood-forming bones of the body. What the system is actually underestimating is that the progeny of these CD34 positive cells have actually also migrated into the tissues where they express luciferase as well. So this is very different than, say, a small molecule or a biologic that would immediately decay over time. Instead, this drug product would expand, migrate, and respond to tissues over time. And so how might we apply these living drugs? The class of genetic diseases, some of which you've heard about today, and sickle cell disease, which you just heard from Ricardo, affects actually hundreds of millions of people worldwide. Many of these diseases are considered rare or orphan diseases because they only affect a small number. But I think this audience is well aware that a single very ill child affects a much larger community of people. Moreover, if one could consider curing a child, one actually expands the number of life years saved. And during the global pandemic, a study came out from the National Economic Burden of Rare Disease study in the United States, which highlighted that both direct and indirect costs of rare disease in the United States exceeded $1 trillion. So these, while individually rare in some, cost both economically and indirectly socially. They pose enormous costs to people in the United States and around the world. So genome editing is a very evocative term. It was invented really in the 2000s. And it implies that we can manipulate the genome, the letters of the genome, in the same way that a word processor can manipulate sentences in a document. And this includes the idea of simply correcting a typographical error. It implies that we can add new sentences to the genome. It implies we can do maybe the equivalent of track changes, where we fix a sentence but leave the unfixed sentence in place. It implies we can replace entire sentences. And finally, it implies that we can add clauses to sentences. And in fact, homologous recombination-based genome editing can do all of these things in a figurative sense. And the way that we do this in my lab, and Ricardo described it as well, is to use a CRISPR-Cas9 nuclease-based system in which we design a guide RNA to recognize the target site in the genome of interest, which recruits, because it is complex, to the molecular scissors, the Cas9 protein, where a double-stranded break is created. And once a double-stranded break is created, it can be repaired in multiple different mechanisms, in multiple different ways, including in a precise repair by non-homologous end joining, which regenerates the target site. And if our RNP complex is still present, another break will be created. And this system, the CRISPR-Cas9 RNP system, is so effective that this iterative cycle of break and repair generally forces the site of the break to go down two mutagenic pathways. One, a mutagenic repair pathway in which there are small insertions and deletions at the site. Or if we provide a donor DNA that has homology to the site of the break, but it contains sequences, nucleotide sequences that change the site of the break in a way we want precisely, we can change, for example, a target site of interest to a target site of great interest, with great representing both single nucleotide changes and changes up to hundreds, if not thousands, of base pairs. So this really gives us the molecular basis, gives us a mechanism to change the genomic sequence as the term genome editing implies, including changing single nucleotides, adding sentences before sentences, inserting entirely new sentences into safe harbors, replacing one sentence with another sentence, replacing one gene with another gene, inserting a gene at the start of another gene so that the genome is rewired, and finally adding new genes at the end of genes, linking them through 2A or irises, in a sense, adding clauses to sentences. I'm using, I guess, the term sentence here to describe a gene cassette. So what I will focus on in this talk is really the most basic version of genome editing, which is to simply change a SNP or a mutation back to a wild type version. And as Ricardo very nicely introduced, the application of this that we've been working on is for sickle cell disease, in which every patient has the same A to T change in codon 6, so the beta globin gene, that when the polar valine is at position 6 in the homozygous state, it predisposes the hemoglobin tetramers to polymerize and converting pliable round red blood cells into the stiff sickle-shaped red blood cells that gives the disease its name. And as Ricardo described, these sickled red blood cells affect the vascular endothelium directly, but also preclude efficient oxygen delivery to all tissues of the body. With a median lifespan for patients with sickle cell disease being in the mid-40s, but the median lifespan for patients born in Africa, where it is estimated about 300,000 patients per year are born, being in the first decade of life. And as again, Ricardo described, while it is a disease specifically of red blood cells, it actually affects all the organs of the body and is manifests most frequently as recurrent pain crises, deep bony pain crises in all the bones of the body. And again, it's like Ricardo and I must have talked to each other, which we have, and to perform a long-term correction of this disease does not simply mean changing the short-lived red blood hematopoietic stem cell, which will give rise to red blood cells for presumably for a lifetime durability. And so, as was mentioned, one way of getting giving a patient hematopoietic stem cells that will make non-sickling red blood cells is to do an allogeneic hematopoietic stem cell transplant from somebody who's an immune match. But as mentioned, it's hard to find patients who are immunologically matched. It's estimated in the US that less than 15% of patients have a good match donor from a sibling. Nonetheless, these allogeneic stem cell transplants have proven that if you can replace the hematopoietic stem cells with those that do not cause sickling red blood cells, you can at least hematologically cure the disease and transform the lives of patients. So the goal then is to replace allogeneic cells by using the patient's own cells, thereby avoiding the challenges of finding a match and the immune and infectious complications from allogeneic transplantation. And the system we use is shown here. It's an ex vivo system in which we will remove hematopoietic stem cells from the patient, and then we will manufacture them by putting the cells into cycle, where the HR-HDR pathway is most active. Deliver the break that initiates the recombination process using a ribonucleoprotein complex in which a purified Cas9 protein. We use a high-fidelity single amino acid variant discovered first by IDT and now sold by both IDT and in GMP quality by Aldevron. We complex this to a single guide RNA that is stabilized, single, and single stabilized and synthetic. So it's made by a machine, and the modifications at the end protect the guide from nuclease degradation. And we deliver this RNP complex to these cells via electroporation. What we learned is that to deliver the donor molecule, however, we could not use naked DNA because, as was mentioned earlier in another session, is that actually naked DNA is a huge stimulant of the type 1 interferon response. It's an immunostimulatory molecule. And so we and others have identified AAV6 as a way of delivering that donor molecule for the recombination process while evading the numerous sensors in the cytoplasm for invading nucleic acids, including TLR9. And using this system, and we also found that delivering the AAV6 immediately following electroporation enhanced or facilitated the transduction of a broader range of primary human cells with AAV6 beyond just what would occur without electroporation, a process that people in my lab called electroporation-assisted transduction. And using this system in a broad array of primary human cell types, including human pluripotent cells, we get on the order of 50 to 80% targeted integration or single nucleotide changes in our cell type of interest. The cell type that stands out is primary neuronal stem cells, which is a bit lower, but these cells divide much, much more slowly, consistent with the idea that the targeted integration reaction occurs in S and G2 of the cell cycle. So using this system and using CD34 cells derived from 6 different sickle cell disease patients and a large team of people, some of whom are shown here, including Annalisa Latanzi, Rasmus Bach, Danny Dever, and Joe Camarena, we could get on the order in the laboratory between 60 and 75% allele correction. You can see that the first few times we were only, quote, only at 40%, but the team just got better and better over time at this process, some of which could be identified, most of which was really just practice. And so I will note to build on what Ricardo said, is that when you have 60 to 75% of the alleles corrected, since we only need to correct one allele, it means somewhere between 80 and close to 100% of the cells have at least one allele corrected. That is, there's conversion from allele correction to cell correction. And as consistent with what Ricardo showed, when we differentiate these red blood cell, these CD34 cells into red blood cells in vitro, we convert the red blood cells from making entirely hemoglobin S to essentially now all non-pathologic hemoglobin A with very little of the pathologic hemoglobin S with the correction frequencies I just showed you. We also did analysis of whether these cells maintain their stem cell potential after transplantation into immunodeficient mice. And here's where the source of the CD34 cells gave us varied results. If we used peripheral blood CD34 cells from sickle cell disease patients that are spontaneously in the circulation, presumably having a significant myeloid bias, the corrected cells engraft quite well into immunodeficient mice with essentially no fall in the frequency of corrected alleles. However, if we use mobilized CD34 cells from non-sickle cell disease patients, what's not shown is we still get the overall correction frequency of 60%, but after engraftment of the same population of cells across many different mice, you see there's high variability of the allele engraftment frequency, with some mice having very high engraftment of gene-corrected alleles and other mice having very low engraftment. What I show here is a blue line around 5% allele correction, which Mark Walters and Robert Ione in a clinical paper suggested could significantly change the course of the disease. And in the red line showing a 15% allele correction corresponding to 20% cell correction from John Tisdale and Courtney Fitzhugh in their study, suggesting that that might be entirely curative for patients with sickle cell disease. Now, when we take peripheral blood mobilized CD34 cells from sickle cell disease patients, generously provided to us by John Tisdale, we find that, again, the correction frequency prior to transplant is around 60%, but the fall in gene correction levels after following transplantation is less than in healthy donors and remains in all of the mice above the sort of Tisdale-Fitzhugh line of 15%. We will note that in our hands and in Luigi Naldini's hands, when you look at the clonal reconstitution of immunodeficient mice following transplantation with gene-edited human cells, it is very oligoclonal, with one to five clones, 1 to 5 human clones generating over 50% of the human hematopoiesis. And so that may be why there is such variability here, because of the stochastic variability of whether you engraft with a gene-corrected cell or not. One thing of interest, of course, is that the cells that do not have a sickle cell allele within the red blood cell lineage, that is, cells that are GPA positive in the marrow, show a selective enrichment for gene correction over cells that where there is no selective advantage for correction, such as in the B cells. So suggesting that, again, you will get a selective advantage in red blood cell development in patients. This is further confirmed in a collaborative experiment done with Adam Wilkinson in Hiro Nakauchi's lab before Adam started his own independent lab at Oxford. And we used what's called the Towns mouse model, in which the mouse globin genes, both beta and alpha, have been replaced by the human alpha and beta globin genes, including a human beta globin sickle cell gene. But before we went to test whether we could correct the sickle cell disease mouse model, what Adam showed is that he could target a GFP cassette into the ROSA 26 safe harbor in mouse hematopoietic stem cells and achieve correction frequencies between, say, 15 and 40%. So not quite as efficient as what we achieve in human cells. But what he could show is in a more or less an autologous model that the gene correction frequency in both primary transplants over a broad range of lineages and in secondary transplant across red blood cells, platelets, myeloid cells, B cells, and T cells did not drop. So again, reinforcing or supporting the idea that in the autologous setting, this drop that we see in the human into mouse xenogenetic setting might not happen. So then, because we have the human beta globin genes in mouse cells, we could directly use the reagents we use to modify human cells. And again, the allele correction frequency is not as high as what we get in human cells because our protocol was not optimized for the smaller mouse hematopoietic stem cells. Nonetheless, we were getting between 15 and 40% gene correction, leaving behind about 15 to 40% of unmodified alleles, with a median of 60% of the alleles developing indels. We could transplant these cells back into the mice, and I just have you focus over here, where at 20 to 24 weeks post-transplantation, you can see across different mice, there's different levels of allele correction in the stem cell or in the myeloid compartment, but a dramatic enrichment of allele correction or the percent hemoglobin A in the peripheral blood, showing again the enrichment of gene-corrected cells. And you can see that even low levels of myeloid correction could lead to therapeutically relevant levels of hemoglobin A in the mice. That is 60% hemoglobin A, which is what is found in most people with sickle cell trait. So we're a little bit ahead of Ricardo and his group, and we're able to scale up this process. And when we scaled up the process to tens of millions of cells, and actually not shown in this is several 100 million cells, our overall HR frequency or gene correction frequency fell somewhat from 60% to the 40 to 55%. But nonetheless, at a cellular correction level, we're again around 50 to 65% above that 20% threshold that Courtney Fitzhugh and John Tisdale has defined. We performed comprehensive or detailed molecular genotoxicity assays. I highlight Dr. Nihar Bhatia's name here because she was an instrumental collaborator with us and unfortunately passed away the day before this paper was published in Science Translational Medicine. And again, we identified across a large number of potential off-target sites, only a single off-target site that had measurable indels above background. This is again using the IDT high fidelity Cas9. This site is intergenic, and when annotated has no functional significance in any cell type, including in the hematopoietic system. We found that using wild type Cas9 in an assay developed by Richard Frock, that there were measurable translocations between the on-target site and the off-target site, but these translocations were reduced by 35-fold using the high fidelity Cas9 to a frequency that we estimate is around 1 in 30,000 cells. We complemented this molecular evidence with an in vivo functional toxicity study that was cleared and reviewed by the FDA, in which a full clinical human dose of gene-edited cells manufactured in the Stanford GMP facility were transplanted across 50 NSG mice, both males and females. And at 16 to 20 weeks, a full necropsy was performed, and there was no evidence of any tumors in any tissues and no evidence of any abnormal hematopoiesis or toxicity from the transplanted cells. I write this all out because it's kind of a negative result, so it's hard to show a negative result, but a positive result for us. So with that, with that efficacy and safety data in hand, we presented it to the FDA. After the usual back and forth, they gave us clearance to initiate a first-in-human phase 1 to clinical trial, which is now open at Lucille Packard Children's Hospital and Wash U in St. Louis, and I believe it's also now open at UAB in Alabama. This trial is sponsored, as I mentioned, by Graphite Bio, but fundamentally, The trial works as many other gene therapy trials, which is to recruit patients globally, evaluate whether they meet the specific inclusion and exclusion criteria. If they do, then to use Plex4 mobilization to harvest their stem cells, purify those stem cells, and use the gene editing process I described in our GMP facility here at Stanford. Cryopreserve them while a aliquot undergoes quality control and analytic testing. And then if the product passes our QC and QA, for it to be released where it'll be infused into a patient who has undergone busulfan myeloablative conditioning to create space for their own autologous cells to engraft into. And we have the patients now being prepared for this process. Now, to end, I just want to sort of touch on the importance, and I think Ricardo mentioned this as well, and this is that while academically it would be exciting to treat a few patients here at our new Packard Children's Hospital, the reality is that these patients exist all over the world, and we need to be able to get these advanced therapies, curative therapies, to patients all over the world. While that is certainly a challenge, I will also say that the one and done cures might be easier and ultimately more cost effective in these parts of the world than, say, a small molecule drug or even a biologic that would have to be given for the lifetime of a patient. And one of the interests I have is to collaborate with people about doing a careful economic analysis of what would it mean to cure a child of sickle cell disease when normally that child would die in the first decade of life. So how do we get there? Ricardo mentioned this to some extent, is we need to streamline the manufacturing process, and I think even more importantly is take it from a open GMP facility with lots of human handling to one that is closed and automated. I think one day we want to prove that this is safe and efficacious in children so that we can mobilize and manufacture healthier and more robust hematopoietic stem cells. It is clear that as sickle cell disease patients age, the inflammatory environment, the stress erythropoiesis, it can damage these hematopoietic stem cells. We need to develop engraftment regimens such that the autologous transplant can be done primarily, if not entirely, in an outpatient setting. And I think there are a couple ways that may get here, and these methods actually could be combined. The first would be to use agents such as antibodies to create niche space instead of having to use toxic chemotherapy like busulfan. And the second would be to somehow endow the gene-corrected cells with a controllable selective advantage so that one could slowly repopulate the marrow with your corrected cells over the uncorrected cells. And we're working on different approaches to that. Nonetheless, we always, I think, want to maintain highly polyclonal stem cell engraftment, because I think if you ask the hematopoietic system to reconstitute from a small number of clones, you are asking for a much higher risk of myelodysplastic syndrome. So with that, I want to thank you for your attention. I've had fantastic people in my lab, collaborators at Stanford in our GMP facility, and we've been really happy to be able to collaborate with people, academic investigators all around the world, including Ricardo and Luis and their team, but also in industry and biotech, where we find that merging expertise, what I've recently learned is a term called asymmetric expertise, can lead to really fruitful collaborations. And I want to thank our funding from both our donors, philanthropic donors, our granting agencies in the US, including the NIH, Doris Duke Charitable Trust, and as well as CIRM, for support in this process. With that, I will stop sharing the screen and look forward to having a nice Q&A with all of you. Thank you very much.
Thank you, Matthew. Thank you, Ricardo. We have some questions, if it's okay for you. I'm going to start with Ricardo, but please, Matthew, please feel free to step in, because I think that several of those questions can be shared. Ricardo, someone asked if what would be the steps that you think that you still have to accomplish to get your study to into a clinical trial?
I think the steps that involve large-scale manufacturing of these cells in a GMP facility. So we are finishing the lab studies, and I think, as Matt described, that's a lot of optimization in this large-scale manufacturing of the cells. I think that's the most important one.
Great, thank you. Matthew and Ricardo, how about your experience? I mean, you both show examples here where you are correcting a point mutation. How do you think will be the efficacy of, instead of correcting a point mutation, of inserting a whole gene, a whole copy of the gene, for example, in a disease where you don't have a common mutation, but you have several different mutations?
Yeah, amazingly, we find that the efficiency of inserting a larger cassette using the AAV system is almost as efficient as correcting a point mutation. We get on the order of 50% insertion of a gene cassette compared to, say, 60 to 80% when we just correct the point mutation using AAV. It is also why I want to give a shout out to Ricardo about testing the SSODNs, because of that could be one place of streamlining for sickle cell disease, but it doesn't apply more broadly to this case where you need to insert a large cassette. I also really want to commend his team for finding ways of minimizing the use of reagents, such as by this compound A. I think that's an area we're also very interested in, because I think streamlining the process also implies minimizing the amount of reagents you use. And I'll ask if I'll take a panelist prerogative whether he's tested the compound A with his SSODNs and seen the same increase, because that would really be very interesting as well.
We haven't yet, Matt.
Probably tomorrow, right? Yeah.
Yeah, it's in the list, but
yeah.
For now, today we're just finishing the in vivo experiment from the compound A, and we got great engraftment with very low MOIs. So in the next week, we will probably know if the editing levels are okay with these lower doses, because this is something that we've observed, and I think you have the same observation, Matt, is that the more amount of virus, the higher the MOI, is much more expensive, and also affects cell fitness. Usually you don't see that clearly in the first few days or in the in the in vitro experiments. But then you go to the in vivo and you see a much lower engraftment. So if you can really reduce the MOI, then you have a much less expensive correction, and you gain in terms of self-fitness as well.
And sorry, we're having a little conversation here, but I will say, I think at least part of that mechanism, I 100% agree, is when you are able to use lower MOIs, you actually activate the P53 response less. And I think the lower you can activate that P53 response, as Ricardo just really nicely pointed out, you increase the health of the fitness of your cells, and so that they engraft better. So, and just to give the audience some clarity, Luigi Naldini's lab and his team have shown that they can get improved engraftment when they specifically inhibit P53. There are some potential risks for that, so we have not adopted that strategy, but the concept is proven.
Yeah, especially when you're generating double-strand breaks, and sometimes some translocation or some large deletions, losing that ability of the cells to sense a DNA damage is quite risky. So if we can decrease the activation of P53, is better than having to silence its activation.
100% agree.
Ricardo, still on that topic, we have a question. If you could talk a little bit more about the SSODN approach, and how is the delivery of this compound along with the Cas9 protein?
Yeah, we just electroporate all together. Yeah, that's the way we do. So it substitutes the AV6. But again, right, it's a DNA donor with a specificity there, so it won't carry the whole gene that you can insert. So it's limited to some types of gene editing, as Matt said before.
Okay, great, thank you. Matthew, if you follow the human CD34 engraftment in mice in sequential transplants, and you evaluate the DNA by the off-target or oncogene NSG panel, do you see any increase in the frequency of mutations? And what about deletions or translocations?
Yeah, no, we have not. That's a great question, a great set of experiments. We don't see enrichment of off-target indels in terms of after following serial transplantation, and in fact, we also have used the TSO 500 panel that Ricardo showed, and showed that even our manufacturing process and culturing the cells out to 10 days, we do not see generation of mutations in cancer-associated genes, nor, and since of course they're not generated, they're not enriched either over time. So we think the papers that were published a couple years ago discussing the risk of P53 mutations were important for us to be aware of, but they were probably a little bit misleading, because what they had done is they had doped in a small number of cells with known P53 mutations, and then measured whether those increased over time as you stress the entire population. I think the implication that CRISPR editing caused P53 mutation is how many of us first read those papers, but that was probably the incorrect interpretation of how those experiments were. It was, if a P53 mutation is created, could those mutations be enriched? But we're not even seeing the creation.
Okay, great. And also for you, we have a second question. Can you expand on how this could be done in an outpatient setting, and next steps needed in the near future?
So, yeah, part of the reason to talk to a really great audience like this is I don't expect that I have all the answers or Ricardo has all the answers, and I hope that the audience maybe will trigger them to come up with some innovative solutions. But I think the pheresis and mobilization process right now in the United States is done inpatient, but really that's just out of a precautionary principle. We don't really see a lot of problems, so that could be done as an outpatient. And I think there are early days now, and I'm lucky to be at Stanford, where the first testing of an antibody to create bone marrow space is being tested. And it's showing some early promising effects. I don't even know if it's version 1 yet, but it has some effects. And my colleague who has been leading the charge on using this antibody to create niche space described that the most toxic effect of the antibody was boredom, because we kept patients in the hospital because we didn't know what was going to happen, and they sat around and they were bored because there were no toxic effects. So I'm not, as I said, I'm not sure this antibody is going to be potent enough, but I think that proves that you can do transplants in an outpatient setting if you have a way of reducing the mucositis, neutropenia, and all the other side effects from busulfan.
Okay, we have another question also for you, Matthew. You also show that in vivo gene editing is viable and successful in his model. How do you see possibility of injecting your gene editing system directly into the bone marrow to treat the sickle cell disease?
I think there's a lot of challenges to doing that. First of all, of course, we electroporate our RNP in, and you couldn't do that in vivo. So then, we heard from Liron, I think, from Intellia, talking about you can deliver the CRISPR-Cas9 on an LNP. But, what we have to be careful of is the LNPs are really designed super well to get into liver cells. And I think they really have to be formulated to target the quiescent HSC, which then leads to my another point, which is that in vivo long-term HSCs are quiescent, which means they're not going to be amenable to HDR. They're going to be amenable to indel-based approaches. Finally, I'll just say that we and others have shown that pre-existing immunity to Cas9 exists. We know that LNP delivery of a foreign protein is a fantastic vaccine. And so threading the needle on getting enough Cas9 expressed in a long-term HSC so that it has its intended effect, but not so long there that you activate the immune system to clear those cells, is going to be very challenging. Now, of course, we're encouraged by the Intellia results and the durability that that you can thread the needle in the liver. Now the question is can you thread the needle in an HSC?
Great. I think we have one more question. Let me just go back here. Okay, so very interesting, the results showing that the circulating CD34 cells, once corrected, engraft more efficiently. Would those cells, assumed to be myeloid skewed, be less efficient in supporting long-term hematopoiesis? Would correction somehow compensate for that potential limitation?
Yeah, I think the person who asked the question is getting at, what we're thinking about as well, is that while are these myeloid biased CD34 positive cells truly long-term HSCs or not? And, this is where I think you do as much as you can in the mouse, human into mouse models, but this is why you have to start a human trial, a small human trial where the patients are carefully monitored to really get a sense of, okay, do you get short-term effects but not long-term effects, and how's that's going to work? I think we could spin our wheels for many, many years trying to decipher, myeloid bias and lymphoid bias and what the subcategories of CD34 cells are. That being said, one of the reasons I like to present our mouse-into-mouse data is it gives more reassurance that the process can work in a long-term HSC that then can engraft through serial transplants in a situation, I think, more akin to what an autologous transplant into humans would look like. I don't know, Ricardo, if you want to add anything to that.
Yeah, I think I agree with that. We see that cells that are mobilized from the bone marrow with Plerixa4 or GM tend to better engraft in the secondary engraftment. Yeah. But it's still, right, we are putting human cells into mice. We don't really know what happens in real humans when the system is more compatible, right? So, yeah, I think it's a fair assessment.
Okay. Okay, so I'm reviewing if we have any additional questions, but I think that we answered, we addressed all of them. So once again, thank you, Matthew. Thank you, Ricardo, for these both great talks and great discussion. I think now Sang is going to give our final thoughts regarding our meeting. Thank you once again for being here with us.
Thank you.
And for this great session.
Okay, now is 5:27 Brazilian time, so we are ending as we programmed it before. So I'm pretty sure that all these presentation discussions about mRNA vaccine and gene editing technologies contribute a lot to us. So I'd like to thank all the speakers again for your excellent contributions. And one message to audience that I'd like to remind that this event was recorded and will be available on demand on ASGCT's website. So if you want to see again or miss a part of this webinar, just go to ASGCT website, okay? So finally, thank you all and see you in the next meetings. Bye-bye.